we are going to talk today about the nucleus of the cell. So we've already discussed the cell membrane, we've talked about the cytoplasm of the cell with all the organelles, and now we're going to talk about the nucleus of the cell. So in this picture you can see this is the nucleus of the cell, and in the middle here, this is a smaller region of the nucleus which is called the nucleus. And the nucleus is like a little storage closet. It's where we store all the different pieces, all the different molecules we need to make up DNA, and then also to make up RNA, which we're going to talk about making both of those in just a little bit. Also, I want you to know that the nucleus has its own membrane around it, and it's usually referred to as the nuclear membrane. Sometimes you might hear people call it the nuclear envelope. That's kind of an old-fashioned term. But this membrane is a phospholipid bilayer, just like we've seen before. You can see it also has these integral proteins, and these integral proteins are called nuclear pores. And information has to be able to go in and out of the nucleus so that the nucleus, the DNA in the nucleus, can communicate its instructions with the rest of the cell. So if you can kind of think of like the DNA is the head honcho of this factory where the cell is building something or multiple different products. But this DNA, it's such a big wig, it can't be bothered to go out onto the factory floor. It just always stays inside its office and likes to keep the door closed, and only certain people can come in and out and talk to this DNA. So we're going to discuss a little bit about what DNA is, what it's made up of, and then we're going to talk about how it controls the rest of the cell. So when we talk about DNA, this stands for deoxyribonucleic acid. When we look at the DNA molecule, now let me actually go to this right here. The DNA molecule is basically made up of 6 different molecules. You have on the sides of the DNA here in this repeating pattern a phosphate, which you see in a P, and then attached to that on either side is a deoxyribose sugar. So you have this phosphate sugar, phosphate sugar, phosphate sugar making up the sides of this DNA, which some people say it's kind of like a ladder because you have the rungs of the DNA in the middle here, and those rungs are made out of what we would call nitrogen bases. These nitrogen bases are your adenine and thymine, guanine, and cytosine. Remember that from your biology class? I teach you all that stuff back then. Okay, so these nitrogen bases are complementary to each other. Adenine always binds with thymine, guanine always binds with cytosine. And so we call these complementary base pairs. Now, a nucleotide is the deoxyribose sugar and a phosphate and a nitrogen base all attached to each other. So again, let's go back to, oops, hang on 1 second, I lost it, there we go. Let's go back to the picture we were just at, and you can see here, this is our phosphate with a deoxyribose sugar with one of the base pairs, and this is called a nucleotide. Okay, so we have these nucleotides making up our DNA ladder. Now, DNA is a really, really large molecule, or I should say a large group of molecules. So let's talk about fertilization and DNA first. So in an egg and in a sperm, you have 23 separate pieces or 23 strands of DNA. And when the egg and the sperm come together, the poor little sperm, it gets to go inside that egg. But as soon as that little sperm gets inside that egg, within just about maybe 5 seconds, that sperm explodes. Now, if you look at a sperm, okay, it's made of a couple different parts. So the sperm has what we call a head, it has a body, and it has a tail. And in this head is where we find all the 23 pieces of DNA. Now the body is actually packed with mitochondria, so it can make a lot of ATP so this little tail can wiggle and swim, because that sperm has a long way to go. Because the sperm is microscopic and it's got to actually swim multiple inches to get to the egg. But as soon as the sperm gets inside the egg, the head of this sperm explodes. And it's a pretty violent explosion, because what we want to happen is we want those 23 pieces of DNA to be pushed inside this egg, so that the 23 pieces of DNA from the sperm come pretty dang close to the 23 pieces of DNA in the egg. Now, as soon as this happens, within maybe, again, another 5 seconds, you start seeing all 46 pieces of DNA vibrate. And you can actually see this under a microscope. It's pretty interesting. And these 46 pieces of DNA begin to vibrate, and they start moving around inside this fertilized egg. And now piece number 1 from mom and piece number 1 from dad will find each other, and they will pair up. And now you have your first pair of DNA, or your first chromosome. And then piece number 2 from mom, piece number 2 from dad will wiggle around until they find each other, and then they pair up. And now you have your second pair of DNA. And this will happen 23 times. All 23 will pair up, and you get 23 pair of DNA in this fertilized egg. So in all the cells of our body, we have 46 pieces, or 23 pair of DNA, except either eggs or sperm. And that pairing up is very important, because it's bringing the child a lot of genetic information from both mom and dad to help improve life. Now, when scientists finally figured this out, they figured out about the 23 pieces and the 23 pair and all that stuff, one of the things that they needed to do was figure out what was the chemical structure of DNA. And I really love and hate this story. There was a scientist, her name was Dr. Rosalind Carter, and she was working at Oxford University in England. And Rosalind Carter was a very brilliant person. And she was headed to be a Nobel Prize winner, because she was figuring out all about DNA. She was the first one to figure out the 6 different molecules that made up DNA. And she was in the process of trying to figure out how they all fit together. But Rosalind Carter made a big mistake. She actually talked to some of her colleagues about what she was doing, and she talked to 2 other scientists who had a lab down the hall from her. Their names were Watson and Crick. And as soon as Rosalind Carter told them a little bit about what she was doing, those 2 rascals decided to do it ahead of her. And they got together and they took all her lab information, and it is even believed that they broke into her lab in the middle of the night and stole things, and they were able to put together the DNA structure. And of course they both won the Nobel Prize from this. Rosalind Carter Pretty much nobody knows who she is, and the really sad thing was, even back then, because she was a woman with a PhD, even though she was working at Oxford, she also didn't even get paid. Women were not allowed to receive a salary. They were supposed to stay home with their kids. And so she was really the brilliant scientist who first studied DNA structure, but Watson and Crick have been given all the accolades for being able to figure out how to put that structure together. Now, needless to say, very smart men, and they did a good job, but they never once gave her credit. In all the books that they read, all the money that they won, each of them won over $150,000, which back in the 1940s, that was a lot of money. They never gave her anything or acknowledged her, which is really quite sad. So one of the things that they were able to figure out is that on the deoxyribose sugar, we're looking at a certain number of carbons here. And they label these 1', 2', 3', 4', 5'. And they found that the phosphate always attached to the 5' carbon on one side, and then the 3' carbon on the other side, which was a very important finding. Now, this finding later became super important because let's say you're a scientist working in, I don't know, Japan, and I'm a scientist working in Australia. And we want to talk to each other about DNA. And maybe we're both studying DNA because we're studying uterine cancer. And we want to look at one of the chromosomes. Maybe we'll look at chromosome number 17. And by the way, we can number each of the chromosomes because they actually have a little bit interesting different length and sort of different shape to them, and so they've been able to number each one of the 23 types of pair of chromosomes for humans. So I can say to scientists across the world, okay, I'm looking at chromosome number 17, but now this chromosome has 2 ends to it. Which end is the beginning and which end is the end? Because like if you're in Japan, you don't read from left to right like they do here or in Australia. Your reading direction is different. So which direction do I read this in? And so scientists had to figure out, oh, holy cow, how do I read the DNA? What direction do I do this in? So here's our phosphate, and it's attached to the 5' carbon. And then we have our base attached, and then we're going to bring another one. This is a phosphate attached to the 3' carbon, and bring in another. And we're attaching phosphates 5' to 3'. And so scientists decided we're always going to read DNA 5' to 3'. And we'll know it's 5' because that's the end that the phosphate is sticking out of. On the other end, which is 3' down here, there is no phosphate. So then we can bring a second chain, the other side of our DNA in, and our bases will bond to each other by hydrogen bonds. Now, notice on the other chain, the phosphate is sticking out down here. So we're going to have the other chain, 5' to 3', in the opposite direction. So it's been said that the 2 chains of DNA are anti-parallel. So we have 5' up here on this side of the chain, 5' down here on this side of the chain. But wherever I'm reading it, doesn't matter which side I'm reading, I'm always going to read my DNA 5' to 3', so I can talk to other scientists about it and they know what the heck I'm talking about. Super duper important. Now, this DNA, it's only made up of 6 different molecules. And these 6 different molecules are really super simple molecules. And all of DNA, all 23 pair of DNA, have a certain number of A's and T's and G's and C's in a certain order, okay? And 99.9% of the order of all of our bases for humans are identical. You and I, as far as our DNA is concerned, we're only 0.1% difference. That's not a heck of a lot. It's very, very little, which is really, truly amazing since nobody looks like anybody in this classroom, really. And personalities are different, and health is different, but yet the majority of our DNA is identical. So that actually makes it really much easier for scientists to be able to take one person's DNA and study it and know that it applies to basically everybody. There's very little that wouldn't apply to the rest of the human race. And so when scientists started studying DNA, that's exactly what they did. They just took DNA from a couple different people and started studying it. And what the big deal was, first question, what the heck does this DNA do? Now we know what it looks like, and we basically know what its shape is. So one of the things that Watson and Crick figured out is how to put it all together, and then they also figured out that it had what they would call this double helix shape, kind of like a spiral staircase type of thing. And so this is an alpha helix. We call it alpha because it actually spins to the right. If the staircase spun to the left, we call it a beta helix. Okay, so now when you're on that $64,000 question or whatever that TV program is, you'll know the difference between alpha and beta. One spins to the right, the other spins to the left. This is an alpha helix. So on the side, we have our sugars and phosphates. In the middle, we have our bases. So Watson and Crick figured out the shape of the DNA, and other scientists came after them and said, so what does this DNA do? And we now know that DNA is like a blueprint. It's an instruction manual that tells our cells how to build protein. So if you can read the DNA, you'll know how to build every single protein in the human body. That includes our enzymes, that includes hormones, every protein that we make. Which also means, since it includes enzymes, it controls the rate of our metabolism, how fast or slow things are done, how much or how little we make of certain things, and that's what DNA is all about. So then the next thing they wanted to know was, okay, so if we now know that DNA makes proteins, could we figure out what's the code, for instance, for the protein that makes hair? Is it a different code for curly hair versus straight hair? And can we tell what that code is? Somebody has breast cancer. Can we figure out what the code is for a healthy breast? And then we'd be able to see if the code was wrong in a newborn child, which would tell us that this child was going to get breast cancer. Could we figure out what the code was for certain diseases? So we know how the code is to control this acid secretion in the stomach. And maybe the code is wrong, and so this person is more likely to get an ulcer. Or maybe we could find the code for the proteins that control the shape of the red blood cells. Because if we find a wrong code, we know this person will have sickle cell anemia. And so that's exactly what scientists have been doing for about the last 40 years, almost 50 years now. So in the early 1970s, a lot of scientists approached the US government and said, hey, look, we want a lot of money to look at DNA, and we think these findings not only will be helpful for humanity, but it'd probably make the government a lot of money. Okay? And so the government said, cool, anything that makes us money we're good with, we'll fund this. And they started what was called Human Genome Project 1. And all Human Genome Project 1 was to take all 23 pair of DNA, start at the 5' end on one side, and figure out what all the A's and T's and G's and C's were, what order they were. That's all they were going to do. Because you know, if you can figure out one side, you know what the other one is. Because if you know it's A, T, G, C, you'll be able to figure out what the complementary code is, right? Okay? So they were going to figure out one side of the DNA, then they would automatically know what the other was. Now, chromosome number 23 in the male is actually a very small chromosome. It's what we call the Y chromosome. It's smaller than the rest. And so they thought, you know what, we're just going to start with a small one, see what we can do with this. Well, it took them a long time because they had to invent new types of instruments and new types of techniques, but eventually they did it. They actually figured out what the pattern was for all the bases on the Y chromosome, which was pretty amazing. And they figured that out in the late 1980s. And just around that time, a little bit later, there came this guy, you know, all of a sudden he became president of the United States, and you've probably heard of him before. His name was like Bill Clinton. And at the same time that Bill Clinton became president, something else revolutionary started to happen. We had like Google and the internet. And Bill Clinton said, you know what? This DNA thing is really important, and I don't want it to be top secret governmental stuff anymore. I want you to take all of the scientific findings you have and put it on the internet. And they did. And holy cow, DNA research exploded. And science around the world were able to see what the American scientists were doing, the new equipment that they had been able to come up with, and everybody and their brother started doing DNA research. Matter of fact, they had a big conference, and they decided that different countries would actually pick different chromosomes, and they would figure out what the pattern of the bases were on a chromosome, so that it happened very fast. And by 1999, they came out with an announcement that the world scientists had decoded all 23 pair of human chromosomes. So that you can get on the internet right now, look at the Human Genome Project, and you can see what all the bases are for all the A's and T's and G's and C's for human chromosomes. As a matter of fact, you may have heard of this. There's a DNA research group called 23andMe, and you can spit in a little tube, and for $150 or $100, something like that, you can mail it away, and they will send you a file of all 23 of your chromosomes with all your A's and T's and G's and C's. It's pretty cool. I did it. It's pretty amazing. So that you have all of your DNA on file. Now what's really cool is they're getting better at this, and there's more people who are willingly giving their DNA, probably eventually to the government, but whatever. They just sent me an email, and they said, we have found about 1,000 cousins of yours. I'm like, holy cow. And it's all over the world, and it goes back to like your 8th cousin, and they're able to map all these people. I have like 150 cousins in California. I don't think I want to know them. The ones I already have, I hardly want to know. Anyway, but it's very interesting to be able to look at the genetic similarities and variances and all. But in 1999, once they were done, they immediately started Human Genome Project number 2. So now in this project, what they wanted to do was they wanted to figure out the individual codes on each of the DNA pairs or on each of the chromosomes that told the body how to make a single gene. Okay, so for instance, let's say I have this piece of DNA, and this is way smaller than what our DNA would be. Okay, so I'm just going to look at one side here. So let's say this is our 5' end, because our phosphate, okay, is coming out here. This is the 3' end. Okay? So I have this DNA, and it's all well and good. I see all the A's and T's and G's and C's. And if I asked you this on a test, okay, so tell me what's the other end say, you can figure that out, right? Because A goes with T and G goes with C, right? Okay, all right. Bio 100 stuff. But now, big deal. What does this say? What instruction is this for which protein? Okay, so now, first thing they had to do was they had to figure out if I've got this big chromosome that has maybe 1 million or more bases on it. Where does the gene start? Or excuse me, where does the code for the protein start? Where does the code for the protein end? And then, once I figure the start and the end, what does this code actually make? So, if I said, okay, this is the code for a protein, if I figure this out, it's a code for a protein, this code is what I would call a gene. This is the code for a single protein. This is a gene. So, as a scientist, we're figuring out these genes, okay? And of course, if you've got 1 million bases on a single chromosome, obviously you're going to have a lot of genes, a lot of separate codes for individual proteins. And they got really lucky when they were trying to figure out the beginning versus the end of these genes. One of the things they found was a pattern. Right before a gene started, they found that there were a whole bunch of A's and T's followed by some G's and C's. And they knew once they found these A's and T's and G's and C's, and I haven't written very many of them, but there would be hundreds of these all clumped together. But once they found these A's and T's and G's and C's all together, they would then know that right here would be the start of that gene. So then you would hope that if I then wanted to find the end of the gene, I would then look for a bunch of A's and T's and G's and C's. And then this would be the start of the next gene, which hopefully this would be the end of the last gene. Yes? Wasn't that easy. Sure enough, this is the start, but the end is a little bit more difficult because they found that it doesn't end right where the next one, the A's and T's and G's and C's start. It doesn't end there. They actually found that there's some of these bases that don't seem to be part of any code, don't seem to be doing anything in particular. For some reason they're there and they haven't quite figured it out yet, and they're still referring to this as just junk DNA. I have a feeling it's not junk. I have a feeling they'll figure out what it really is. But for now, they're just calling it junk. So it's been a little bit difficult. They're still working on where do genes end. They got the beginning, we're good. But exactly where do they end? This has still been a little bit tough. But scientists have found a lot of these genes. Like for instance, we know that on chromosome 17, there are multiple genes that control the proteins that make the human breast. And so they can look at these genes and know what the correct code is versus what a wrong code is, and then be able to say if somebody has a chance of cancer. They've been able to find the genes that control a whole bunch of other things to be able to tell us if children are going to have certain genetic diseases or not. Matter of fact, they now are looking at even more than just diseases. They're finding genes that control the color of your hair, the color of your eyes. They're finding genes that control certain traits in a person. Like for instance, does it run in your family to have a gap between your teeth? Does it run in your family to be lactose intolerant? They're looking at a whole bunch of different traits in individuals. And so these genes are slowly coming out, and we're slowly learning more and more about them. Now, by the way, at the same time scientists were learning about human genetics and human DNA, there were other scientists that were also studying animals and plants and insects and you name it. They were studying the DNA of it and trying to map the genetics of all these different creatures. And they've been very successful. Matter of fact, we've been super successful. Maybe way too successful. Because many scientists have been able to map the genetics of many animals and plants much faster because they're a lot more simple than humans. And now this also means that we can manipulate the genetics. We can change them, and we can modify the genetics. And now we have two different types of organisms in the world. We have natural organisms, or what we call organic, and we have GMO, or genetically modified organisms. And the problem so far has been that genetically modified organisms appear for the most part to be genetically hardier, stronger than non-genetically modified. So that let's say we genetically modify a plant. If the pollen from that genetically modified plant blows into an organic original plant field, those plants will all become genetically modified. It won't go the other way. The organic originals are not as strong, and they will not blow into the genetically modified plant field and turn them back into original. It doesn't happen. Which is a little scary because there is no control over this. There are no ethics boards to control these scientists. There is absolutely zero control over genetic modifications in the United States. Now, Europe, Canada, many other countries have stopped this type of genetic modification. As a matter of fact, they will not even allow American food products into their country because they are afraid that it will start to modify their food products as well. And a lot of the modifications that have happened to our food products, for instance, are not really turning out to be all that good. So I'll give you an example. Corn. It was genetically modified some years ago. It was actually the first genetically modified plant. And what they did, they thought, this is really cool stuff. What we're going to do is we're going to actually put into the corn plant genetics that will allow the corn plant to poison bugs. So that when the bug eats this corn plant, the corn plant will now produce a protein that will make the bug's intestines explode. Now this should save the farmer a lot of money because now they don't have to use any kind of pesticide on their plants, and it should make us more healthy. That really was the thought. Until what we found was that when we eat this genetically modified corn plant, it also makes our intestines explode. And now we have a brand new disease we've never seen before in the history of man called leaky gut syndrome. And leaky gut syndrome means that you have these microscopic explosions that have happened through your small intestine so that you have these pinpoint holes in your small intestine. And now food, which is not supposed to be able to get out of your small intestine until it's much tinier, okay? It's been broken down more. Larger amounts, larger pieces of food can now enter into your body, get into your bloodstream, go to places it's not supposed to be able to go, and create a reaction. And what happens is when that happens to us, our immune system goes bananas and starts attacking this food stuff that's floating around that should not be in there, and the immune system starts releasing chemicals that our body reacts in a couple different ways. One, we react as if we have allergies. Or two, we react as if we have an autoimmune disease. So that now you see children with allergies like you've never seen them before. I mean, for instance, my grandkids, 2 of them, they go to a school, and their dad is from Egypt, so he doesn't know this stuff, and he made a peanut butter and jelly sandwich for their kids' lunch and sent the kid to school. And they were actually kicked out of school because you can't bring peanuts. Because there are kids now that have peanut allergies so bad that if they just smell it across the room, they'll go into anaphylactic shock. We've never seen this before. This is not normal. And these genetically modified foods, here's the other thing. They were never ever tested before they were given to us for us to eat. They were just made, they were just modified, and then they were given to all of us to consume, and nobody did any kind of scientific testing to see what the heck would happen to us if we ate this stuff. And now the state of California has agreed to genetically modify animals. And so eventually what they're hoping is they're hoping to have one genetic source for all of your cows. So that all of your cows look the same, taste the same, have the same genetics. And they're trying to get one genetic source for all of your plants. So that all the foods we eat are from the same genetic source. So every tomato is the same genetic source. Every corn is the same genetic source. As a matter of fact, there's only one company doing this, and this is a company called Monsanto. And Monsanto is very powerful, okay? And the government has given them the green light to go ahead and do this, and Monsanto is not stupid. They are a smart company. Matter of fact, what they are doing is they are now going around and buying up all the organic seed companies and putting them out of business. So that eventually the normal seeds will not even exist. And you will have to buy what they sell.
But why? What is the point of that? What's the point of it?
Serious money. Serious money. That's the point of it. It's to be able to control the food source. And they've been given the okay to do that. The Supreme Court actually passed a law that said they're allowed to do that. You're allowed to patent food sources. You never were able to do that before. Ever, ever, ever. It used to be colleges and universities did a lot of studying on how to grow better plants, and then once they got this really awesome plant growing, they would freely, for free, give the seeds to farmers. Okay? Now that is not the case. As a matter of fact, Monsanto is so stingy, okay? If, let's say I'm a corn farmer, all right, and I want to plant 100 acres of corn, and I have to buy Monsanto's corn seeds. For 100 acres, it'll cost me about $30,000 to plant corn. Now, it wouldn't be all that expensive if, once my corn grew, I could save some of those corn, and I could dry out the seeds, and I could plant those seeds next year. Monsanto says you're not allowed to do that. Every year, I have to buy seed from them.
So is that like a private-owned company?
Yes. private owned.
That's great.
Now I will tell you that they are also getting into genetic modification of humans. It's closer than you think. We've already genetically modified animals, and a lot of people are doing this. So for instance, in Japan, a couple years ago, there was a scientist who said he had a big demonstration of a genetically modified animal. And he walked out on television with this really cute white cat, fluffy white cat. Set the white cat down, walked out, turned off the lights, turned on a black light, and the cat was glowing in the dark. What he had done is he had taken the fluorescence of a jellyfish and he had spliced that into the egg of the cat and then artificially inseminated mama cat, and now you have a glow-in-the-dark kitty cat. They've since done this to monkeys and to rabbits and to multiple other types of animals.
At least you know it's coming.
And so do the coyotes and everything else, and so yeah, you don't have to worry about it.
So that cat is stronger than
Potentially.
Yeah.
But now that cat, if it reproduced, all of its kittens would also glow in the dark. So now what you've done is you've introduced a genetic variant into the world that potentially you can never get out of. And what does that do into your ecosystem? I don't know. I have no idea. When you produce a genetically varied plant or a genetically varied animal, what does it do to your ecosystem? I don't know. Now, see, here's the thing. Plants also change the ground. They change the groundwater. So Monsanto came out with a genetically modified cotton plant. Excuse me, this one actually was a rice plant. And they wanted other countries to buy into this, so they sold to South Africa, they sold to India, told them it was going to be bigger, badder, stronger, they'd be able to produce more. And then what happened was the plant, as it grew, became very acidic and destroyed the soil. And many, many, many farmers have no land anymore. And it will be years and years and years before the soil will be able to heal itself. And you can look online about this. In India, they had massive suicides of farmers because in their culture, if something like this happens and you can't provide for your family, you have no alternative except to kill yourself. And now you have all these farmers who are dead. Now who will feed the people? And what land will they use? It's changing a lot of things. And there's a lot of people who are now very much against it, which is why you see more organic things coming out, because organic means not only there's no pesticide, there's no herbicide, it's also not genetically modified, or at least it should be, okay, as much as you can trust what they're telling you. That's why I also recommend you try to find a farmer and buy from them, and I know that's harder and harder to do in Southern California. There aren't as many farmers. I mean, we're getting food from Chile and from, you know, all over the place instead of around home, because we've built buildings instead of feeding ourselves, which was a little bit on the crazy side.
If they're genetically modified from an original DNA, could they be genetically modified and go back to that original DNA?
They could, yes, they could. They absolutely could. But you have to stop Monsanto. Good luck. Now, there have been a lot of lawsuits around the world against them. And in those countries, they've won. There have been a lot of lawsuits in the United States against them, and not a single lawsuit against Monsanto has stood. Monsanto's won every single one. They're such a powerful company in the United States.
Because of money?
Oh, yeah. Yeah, there's lots of money. They're very entwined with the government. Very entwined.
Lots of lobbyists.
Yeah. Lots of people from government work for Monsanto. From Monsanto, they go to work for the government. It's a very smart company. Now, of course, you got to think about this too. You know, are you totally against genetic modification? because you could heal a lot of people with it. It's real simple to do. Super duper duper simple. And it is already happening. Okay? They are already modifying humans. It's already happening all over the world, including the United States. So what you can do is you take the egg, you take the sperm, you put them in a petri dish. You get that sperm to go inside the egg and explode, and you get all 23 pair of DNA to line up. And as soon as you see them lined up, because you can, again, just see it under a microscope. It's not that hard. Just a regular microscope like that. As soon as all 23 pair are lined up, you freeze it. And now you can work with that cell all you want. It won't split, it won't grow. And one of the things that I can do is I could defrost it, and I could take a little glass pipette and put it through the cell membrane. It won't hurt it. The cell membrane is kind of sticky. It'll stick around the glass pipette, and I can suck out all 23 pair of DNA. And then I can take those 23 pair of DNA and I can put them into what's called a microarray. And this instrument can read all the bases in all 23 pair of DNA and tell me what the bases are on every single chromosome from 5' to 3'. And if I know what all the genes are, I know what the codes are, I know what they make, I could then cut certain sections out that maybe were no good, wrong code, and I could put in the correct code. And now I put that DNA that's been totally corrected back into the egg and put that fertilized egg by in vitro fertilization into mom, or they're making an artificial uterus, by the way. I put it in there. Mom never loses her figure. And watch the baby grow. And now this child no longer has any genetic defects. Hard to be against that, huh? But then I could also build a baby.
Like a workshop?
Uh-huh. Just like build a bear, I could build a baby. I go in and say, okay, I want my child to be this tall. I want them to have this color eyes, this color hair, this kind of skin. I want them to have this kind of personality, which, by the way, is genetically controlled. I want them to have these many neurons because I want them to have more neurons, because the more neurons you have, the greater your potential for intelligence. Okay? So let's start them off with more neurons. I want them to be this strong or not so strong. You name it.
Excuse me, Dr. Harvey.
Yes?
Is when you do that in a human, it's also the same as cloning?
No. No. Cloning is taking the identical DNA and making an identical person, like an identical twin.
They don't have to remove anything.
No. No, you're a clone. He has an identical twin. So they are true clones. So how many of you would be willing to build a baby? I want him to look like this, I want him to be like this, I want him to have more musical talent. You wouldn't?
It's crazy.
No.
It's gonna be expensive.
It wasn't.
It probably would be at first, but it is becoming more and more inexpensive. So if it wasn't expensive, would you be okay with building a baby?
I know.
Now, by the way, when they first came out with penicillin and new penicillin killed multiple different types of bacteria and people were healed from it, do you know what most people's response was? If God wanted them to live, they would live. We should not be using this stuff. It's unnatural. Now, of course, none of you believe that now. You get sick, you take penicillin or whatever.
It could be beneficial for humanity.
Yeah.
In certain ways, though.
Yeah, like what?
I think the interesting thing would be the interaction between the organic humans and the modified ones and how that would play out.
Do you know that that is a book called 1984?
Really?
And there's another one called Brave New World. Read them. They're all about organic versus modified humans. It's very, very interesting, especially Brave New World. That's a really, really good one. Because one of the modified humans, the test tube breaks. And so he becomes really scrambled. A very interesting story, but it was written in the 1930s.
And neither book ended well.
Yes.
I think I took the excitement out of everything too. I think.
Yeah, it's the struggle with the fun. I mean, if you just made perfectly.
Come on, they're going to be your kid. Don't you want them to look the best, have the best intelligence, have the greatest personality? No.
They should suffer and then they should get through it.
Okay,
and
but
be better.
see, you know what?
Yeah.
They
Dr.
should-
Harvey, that's very unethical. I don't want to.
It
I
is?
want to know what they have
For me,
what their babies will look
But
like.
some they don't.
I mean, it'd be nice to have no kids that were like born with Down syndrome,
Yeah, but I mean,
you know, stuff like that.
Come on, you don't want to have a child who would be like the best football player ever.
But all of our
Everybody
kids
else
will
is
be the
busy
best
too.
They wouldn't be more special. There is no room for diversity.
That is a problem. You would have a lot of people who are very similar to each other. And by the way, ladies, there'd be a lot more men in the world. Because most people want a male child. The majority of the
Until
world
they have
over
one.
would prefer,
Yeah.
the majority of the world over would prefer male
Hey,
children.
I'm taking offense to that over there.
And so you could build that child to be what you wanted them to be. There's a lot, you may say no, because you're quite more educated than a lot of people in this world, but let me tell you, there's a lot of people who would say yes.
Oh, my son said it's cool.
Yeah.
And he said, you're crazy.
Absolutely.
Because they don't have any idea.
And wouldn't you like this? Wouldn't you like to be able to take unknown sperm and unknown eggs and put them together and build them in an artificial uterus and raise those children to be your soldiers so that none of your kids had to go to war?
It's like Star Wars.
And now we can genetically modify them that they'll be stronger and they can last longer. And we can make some of them smarter and some of them dumber. The smarter ones could go like into the Air Force and the dumber ones could be our Marines. Hey, look, I'm a Marine Corps brat. Hoorah! My father was a colonel. Okay, I know what a jarhead is and a bullet stopper. That's called a Marine. My brother is a major right now in Pendleton, so I got a lot of experience with hoorah Marine Corps, okay? It's true, and they would tell you the same thing. They're out there to be bullet stoppers. They'll take that bullet. That's what they're trained to do. They're trained to be the first one out. They're trained to be the first one to die. 100% true. And that's what they're proud of. They're proud of being bullet stoppers. And you don't have to have a lot of brains to be a bullet stopper. You just have to be big, bad, and fast. And take orders.
Sure, yes, sir.
Sir, yes, sir. That's right.
Military.
So why not? Then you wouldn't have anybody worried about is my son that died.
Deep inside, a lot of people like all those doing those things. Psychologically, they like.
Well, we would let them if they really wanted to do that.
Just going together.
Yeah, because anybody in my family would want to be,
Yeah.
you know, one of those dummies that did it. Yeah. Oh, I knew you missed me.
Yeah. Come in here.
All the time. All the time. It's very interesting, isn't it? You guys, it's really funny because when I very first started teaching, which was in 1993, probably before some of you were born, I taught about this, and this was way in the future. I mean, Human Genome Project 1 wasn't even done yet. And now, this is already happening. You're already eating genetically modified foods. If you're not buying something that says organic, it's genetically modified. And we're talking fruit, vegetables, meat. It's already there. But you're not waiting for the future to occur. I mean, you already have glow-in-the-dark kitty cats, okay? By the way, could you not also do this? Could you not genetically modify humans to also be part animal?
I could be one.
Planet of the Apes, dude. Planet of the Apes.
Yo, I want some wings now.
That's exactly right.
Yes.
By the way, somebody's trying to genetically modify a horse so that a rhinoceros horn will come out. You got your unicorn. Why couldn't you take condor wings and put it on a human?
Not gonna lie, that stuff is actually sounding pretty cool.
You get to fly.
Yeah, you never be late.
We will not be late.
Drop a hamburger or something.
See, some of it doesn't sound all that bad, let me tell you.
Maybe like Waterworld and get some gills.
See? Why not?
You never can come back up.
Teeth
there,
like a
man.
shark? Why not?
A mermaid so that we can breathe under the
Ooh,
sea.
mermaid.
Yeah.
Part dolphin, part human. Mhm. It sounds really far-fetched, but it actually is not all that far off. You are looking at a lot of legislation right now going through to try to genetically modify humans and make it legal. And this will be the country that does it first. I mean, we are truly, truly trying to make an artificial uterus right now. So women can just watch their baby grow.
So no need for surrogate mothers.
Huh?
We do not need a surrogate mothers anymore.
No, you don't need surrogate mothers. You have an artificial uterus. Now, by the way, here is one thing to remember. When you do start genetically modifying plants and animals, you can start to lose some of the immune protection that they've made over the centuries. Some of that adaptability to the environment can also be lost. And especially if you're modifying plants and you have to replant every year the same seed. That seed hasn't learned to adapt to its environment. And the viruses, the fungi, the bacteria around it have. So when you plant that older seed, you don't have the same virus now when you plant the seed again, because that virus now is more powerful to kill that plant. So you have the potential also to produce famine. The whole idea is these are supposed to be bigger and badder, but then you lose the immune system part of it. And will we lose that in humans too, depending on the changes we make? Well, we don't know. Nobody's going slow with this because scientists are totally thinking this is cool, and they are just like running ahead like a bullet train. Because this is just so awesome and they just want to see what happens. Let me tell you one thing. When you have a scientist that's enthused, you so need to control them. They need to have like something on their neck and you just rein them in, because they're just going to do it, and then go, oh shoot, maybe I shouldn't have done that. Like the people who created the atom bomb. They were very upset afterwards that they had done that. They had no idea what they were actually making. Nobody in the world had seen it before. And the majority of scientists, if you read the books that they wrote, they said that they basically never slept again.
A lot of people died of that.
So when legislation comes up for you to vote on controlling genetic modifications, and it will, and it has in the state of California, for instance, a couple of years ago, it came out, and what they wanted to do is they wanted to make sure that all companies had to label the foods that they made genetically modified. And the state of California voted no. Because Monsanto had these great commercials that made everybody think that it would hurt the farmer. It doesn't hurt the farmer, it hurts Monsanto. Monsanto is the farmer. Monsanto controls most of the farms in the United States. There's not a lot of small farms anymore. They're all corporate farms. So when Monsanto said it would hurt the farmer, they meant actually, well, it'll hurt us, so we don't want you to do this. So when those legislations come out, you've got to think about this biologically and try to gauge the long-term impact to the world and what it's doing to us. Really very important. Any comments?